Let's begin chapter 25, the history of life on Earth. Past organisms were very different from those that are now alive. The fossil record shows macroevolutionary changes over large time scales. For example, the emergence of terrestrial vertebrates, the impact of our 5 mass extinctions, and the origin of flight in birds. So here is a picture of 2 dinosaurs, which you can see are very different from the organisms that are now alive today. So let's talk about the conditions on early Earth that made the origin of life possible. On early Earth, there were chemical and physical processes that produced very simple cells through a sequence of 4 stages. First, there was abiotic synthesis of small organic molecules. Then there is the joining of these small molecules into macromolecules. Then the macromolecules are packaged into protocells, which is the origin of the self-replicating molecules. So let's talk about abiotic synthesis of organic compounds on early Earth. Earth was formed about 4.6 billion years ago with the rest of the solar system. In the early atmosphere, there was water vapor and a variety of other chemicals such as nitrogen, methane, ammonia, and other chemicals which were released by volcanic eruptions. During that time, volcanic eruptions were happening everywhere. In the 1920s, Alexander O'Pardon and John Holden hypothesized that the early atmosphere was a reducing environment. A reducing atmosphere contains reductants or reducing agents that are saturated with hydrogen ions and are able to reduce other molecules. Remember our oxidation and reduction from chapter 8. If you don't remember it, go ahead and look it up. Now, a predominant experiment was performed in 1953 by Miller and Urey to mimic the conditions of early Earth. Their experiment showed that abiotic molecules could be used to create important biotic compounds thought to be necessary for the origin of life. So they showed how it was possible for non-living molecules to be used to create living molecules. So Miller and Urey set up a closed system to simulate the conditions thought to have existed on early Earth. They created a reducing atmosphere which consists of hydrogen gas, methane, ammonia, and water vapor. They then mimicked lightning by discharging sparks. Miller and Urey collected samples to see if there were any organic molecules. They identified a variety of organic molecules, including amino acids such as alanine and glutamic acid, which are common in proteins such as organisms in our cells. So they showed how organic molecules, which is the first step to the origin of life, can be formed from a strongly reducing atmosphere. Although Miller and Urey type experiments demonstrated that organic molecules could have formed with various possible atmospheres, the evidence is still not yet convincing that the early atmosphere was in fact reducing. So instead of forming in an atmosphere, there is another hypothesis that says that the first organic compounds may have been created near volcanoes or deep sea vents because of all the energy that is produced. Now, amino acids have also been found on meteorites. There is a whole group of people that believe in something called exobiology, which is that the first organic compound came from space on a meteorite that struck Earth and left behind these organic molecules that promoted or prompted life. So the next step in the production of simple cells was the synthesis of macromolecules. So small organic molecules can be joined when they are on hot sand, clay, or rock. The heat provides the energy or reaction source to join them into macromolecules. So once the macromolecules are formed, they are packaged into protocells. Protocells are essentially just a membrane wrapped around some fluid. So they are fluid-filled vesicles with a membrane-like structure. In the vesicles, metabolic reactions can occur. It provides a separate internal chemical environment for reactions to take place. Now, it is important to know that replication and metabolism are key properties of life and may have appeared together. Evidence of the formation of protocells is when lipids and other organic molecules are put in water and can spontaneously form vesicles with a lipid bilayer. You then can add clay, which can increase the rate of formation. Remember, hot clay is the place where small organic molecules are joined, which we can see here. As we increase the amount of clay, it increases the amount of production of these protocells or vesicles. Now, we believe that life originated as an RNA world. So the first genetic material was probably RNA and not DNA. We think that it was probably RNA and not DNA for a couple of reasons. First, RNA can be self-catalytic and create molecules called ribozymes. Ribozymes are enzymatic RNA molecules that can catalyze reactions, including their own breakdown and their own formation. So they are self-splicing and also can make complementary copies of their own sequences or other short stretches of RNA. So they are catalytic in the formation of RNA, which is extremely important. Now, early protocells with self-replicating catalytic RNA would have been more efficient at using resources and would have increased in numbers through natural selection. So natural selection has selected for self-replicating RNA molecules. RNA molecules that were more stable or replicated more quickly would have left more descendant RNA molecules. So again, the early genetic material might have formed an RNA world. So vesicles with RNA capable of replication would have been the protocells. RNA could have provided the template for DNA, a more stable genetic material. So let's go into concept 25.2. Fossils are a major source of information for us. Fossils used to be largely the only thing that we had to provide proof of things that existed a long time ago and the changes. Remember, sedimentary rock are deposits that have layers called strata, which are rich in the source of fossils. An important piece of information that can be gained from fossils that we dig up is the comparative age or relative age based on depth. The deeper something is buried, the older it is. It is that simple. So there are few individuals that have ever been fossilized and even fewer that have ever been discovered. The reason why is because the fossil record tends to favor species that existed a long time ago, were abundant and widespread, and had hard parts so the fossils could be formed. And fossils tend to be discoveries that can be a matter of chance or prediction. For example, paleontologists found an early terrestrial vertebrate by targeting sedimentary rock from a specific time and environment. So they were looking at a specific time and a specific environment to find something. So as I said, sedimentary strata can reveal the relative age of fossils, but the absolute age of fossils can be determined by radiometric dating. Radiometric dating is done by measuring the half-life of the molecule. So a parent isotope decays to a daughter isotope at a constant rate. Remember, isotopes are atoms with different number of neutrons. Each isotope has a known half-life, which is the time required for half of the parent isotope to decay. So radiometric dating is done by measuring the half-life of a molecule. Now, it is usually done with carbon-14. So we are measuring the time it takes for carbon-14 to lose a neutron to carbon-13, and then to lose another neutron to carbon-12, and so on and so forth. Here is a diagram of a radiometric dating. So radiocarbon dating can be used to date fossils up to 75,000 years old. Now for fossils older, some isotopes can be used to date sedimentary rock layers above or below the fossils. Mammals belong to a group of animals called tetrapods. The evolution of unique mammal features can be traced through gradual changes over time. So we can see here from the structure of each skull how the evolution of mammal features can be traced through gradual changes over time. So let's get into concept 25.3. The geologic record is divided into 3 eons: the Archean, the Proterozoic, and the Phanerozoic. The Phanerozoic encompasses multicellular eukaryotic life. The Phanerozoic is divided in 3 eras: the Paleozoic, the Mesozoic, and the Cenozoic. Here is a geologic record that shows the different eons, how long ago they were, and some important events in the history of life. Major boundaries between geologic divisions correspond to extinction events in the fossil record. Now, some like to describe the geologic record as a time clock divided by major events. It starts with the origin of Earth, then the first living things are the prokaryotics. They include the archaeobacteria, which are able to survive in harsher environments because there is little to no oxygen. The prokaryotics then were the cause of the release of oxygen into the atmosphere, which was then followed by multicellular eukaryotic organisms, then animals followed, and then land plants, and finally a tiny little sliver, which is us, humans. So let's go ahead and talk about the first things, prokaryotics. The oldest known fossils are called stromatolites. They actually look like rock, but are actually bacteria that has ingrained in part of the sediment. The oldest one is dated back to 3.5 billion years ago. Prokaryotics were Earth's sole inhabitants for about 1.4 billion years, from 3.5 billion to 2.1 billion years ago. Here is a picture of the stromatolites. As you can see, they look like rock, but they actually are bacteria embedded into the sediment. So let's talk about the beginning of atmospheric oxygen. Most atmospheric oxygen came from something that is biological. Oxygen can be produced by oxygenic photosynthesis that then can react with dissolved ions and precipitate out to form bands of ion formation. So about 2.7 billion years ago, oxygen began accumulating in the atmosphere and rusting ion-rich terrestrial rocks. This was known as the oxygen revolution and was from 2.7 to 2.3 billion years ago. Now, this caused the extinction of many prokaryotic groups. Now, some of the groups survived and adapted to using oxygen and cellular respiration to harvest energy. Here is a graph that shows the percent of atmospheric oxygen over the course of time, and you can see from the graph that between 2.7 and to 2.3 billion years ago is when the oxygen revolution took place. Now, the first production of oxygen was 3.5 billion years ago, and this was from cyanobacteria that performed oxygenic photosynthesis. The later increase in the rise of oxygen during the oxygen revolution might have been caused by the evolution of eukaryotic cells containing chloroplasts. So let's talk about single-celled eukaryotes. The oldest eukaryotic cells date back to 2.1 billion years ago. Remember, eukaryotic cells have a nuclear envelope, a mitochondria, an endoplasmic reticulum, and a cytoskeleton. Those are the key organelles of a eukaryotic cell. Now, the endosymbiont theory proposes that the mitochondria and the plastids, which are a large family of organelles that include the chloroplasts, were formerly prokaryotic cells living within a larger host cells. Endo means with inside, thus it is a symbiotic relationship inside. And an endosymbiont is a cell that lives within the host cell. The prokaryotic ancestor of mitochondria and plastid probably got into the host cell as an undigested prey or as an internal parasite. So they were taken in by endocytes and not digested, but survived and lived on. Now, serial endosymbionts supposes that the mitochondria evolved before plastids through a series of endosymbiotic events. So, you can see here that the prokaryotic is engulfed, evolving into a nucleus. Then another prokaryotic is involved, and the mitochondria evolves. Then, through a sequence of other endosymbiotic events, the photosynthetic prokaryotic, like cyanobacteria, is engulfed, and the plastids evolved. So you can see that there is serial endosymbionts, that the nucleus first forms. It is then followed by the mitochondria, followed by the plastid, through a sequence of endosymbiotic events. Now, there's significant amount of evidence supporting an endosymbiotic origin of the mitochondria and plastid. Here is a small list of some. First, there is an inner membrane for both the mitochondria and the plastids that is similar to the plasma membrane of prokaryotics. Another key piece of evidence is the division is similar in these organelles and in some prokaryotics. Next, these organelles transcribe and translate their own DNA, and their ribosomes are more similar to prokaryotics than they are to eukaryotic ribosomes. So these are some evidence that supports the endosymbiotic origin of the mitochondria and plastids. So the evolution of eukaryotic cells allowed for greater range of unicellular forms. A second range of diversity occurred when multicellular evolved and gave rise to algae, plants, fungus, and animals, including ourselves. So let's talk about multicellular eukaryotics. Let's talk about the earliest multicellular eukaryotics. The oldest known fossil of a multicellular eukaryotic is about 1.2 billion years ago, and they were very small algae that lived. We can date the common ancestor of multicellular eukaryotics to 1.5 billion years ago. Now, there is a hypothesis called the snowball Earth, which suggests that periods of extreme glaciers combined life to areas around the equator and deep sea vents from 750 to 580 million years ago. The Adirondack biota were an assemblage of larger and more diverse soft-bodied organisms that lived from 575 to 535 million years ago. Now let's get into animals. Another major or important episode in the geologic record is the Cambrian explosion. It is attributed to a majority of the animal species that existed today. It is unsure how this happened, but at a large time we got a large number of the major phyla of animals. It provides evidence also of prey and predator interactions. Here's a cladogram that shows the appearance of selected animals group. So we can look at the molecular evidence to see when an animal phyla began to diverge, which means looking at the number of differences genetically in the DNA or the protein structure. We can see that the animal phyla began to diverge as early as 7 million years to 1 billion years ago. This is where we see differences add up. Now, there were fossils found in China that provide evidence of modern animal phyla 10 of million of years before the Cambrian explosion. The Chinese fossils suggest that the Cambrian explosion had a long fuse. So here's a fossil from China that is 570 million years old and is believed to be animal embryos. Figure A shows the embryo at a 2-cell stage, and figure B shows a later stage. So let's get into the colonization of land. We know that fungus, plants, and animals have been on land for about 500 million years. Vascular tissue in plants transport material internally and appeared about 420 million years ago. There is a symbiotic relationship between plants and fungus that still exists today, which likely has helped them colonize land together. Arthropods and tetrapods were most widely spread and diverse land animals. Tetrapods evolved from the lobed fin fish about 365 million years ago. Now let's start concept 25.4. The history of life on Earth has been a rise and fall of many groups of organisms, and it all depends on the speciation rate and extinction rate within a group. At 3 points in time, the land masses of Earth have formed a supercontinent. 1.1 billion years ago, 600 million years ago, and 250 million years ago. According to the theory of plate tectonics, Earth's crust is composed of plates floating on the Earth's mantle. Here is a picture of a cutaway view of the Earth. You can see that the Earth's crust, which is composed of plates, is floating on the Earth's mantle. We know that the Earth's crust is not fixed and tectonic plates constantly move slowly through a process of continental drift. Oceanic and continental plates can collide, separate, or slide past each other, which causes earthquakes, volcanoes, and mountains and islands to be formed. Here's a map of the major tectonic plates and their boundaries. Now, everyone has probably heard of Pangaea. Pangaea is a supercontinent that was present about 250 million years ago. It had many effects. First, it caused a deepening of the ocean basins. Then, it caused a reduction in the shallow water habitats. It also caused colder and deeper climates inland. Here is a diagram of the supercontinent Pangaea and how continental drift took place, forming the layout which we currently have today. Now, continental drift has many effects on living organisms. First, the continent's climate can change as it moves north or south. Second, separation of land masses can lead to allopatric speciation. Remember, in allopatric speciation, a population is divided into two subpopulations by a geographical barrier and can be formed into two different species. Now, movement of continents can reflect the distribution of fossils and living groups. For example, the similarities of fossils in part of South America and Africa is consistent with the ideas that these continents were formerly attached. Now let's talk about extinction events. Fossil records show that most species that have ever lived are now extinct, and their extinction was usually caused by the change in the environment. Now, at times when the rate of extinction has increased dramatically, it can cause what we call a mass extinction. A mass extinction can result in disruptive global environmental changes. There are 5 major extinction events, and more than 50% of the Earth's species have become extinct during them. Figure 25.15 shows the mass extinction events and the diversity of life. You can see where the mass extinction events occurred because there is a high peak in the graph, as you can see here, 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 and here. It also shows you down here with little arrows our 5 major extinction events. Let's talk about one of the mass extinctions called the Permian extinction. It is well known because it caused the extinction of 96% of the marine animal species and also 8 out of 27 insect species. So tons and tons of animals went extinct in this mass extinction. The Permian extinction happened during the Paleozoic and the Mesozoic eras, about 251 million years. A number of factors may have contributed to the Permian extinction. First, there were intense volcanic eruptions in what is now Siberia, and global warming resulted from the emission of large amounts of CO2 from the volcanoes. There was also a reduction in the temperature gradient from the equator to the poles. And another factor that might have contributed to the Permian extinction is the oceanic anoxia, which is a low amount of oxygen that may have been caused from the reduced mixing of the ocean's waters. Another mass extinction is the Cretaceous extinction, which is the dinosaur extinction. Here there was a large number of deaths, both marine and terrestrial species, including dinosaurs. It happened 65.5 million years ago and separates the Mesozoic from the Cenozoic eras. The Cretaceous extinction is said to be caused by a meteorite impact that caused a release of debris into the atmosphere, which blocked the sunlight, which affected photosynthesis and the food chain. Here is the location where the meteorite is thought to have caused the Cretaceous extinction, which is located in Mexico. Now, is a 6th mass extinction underway? Scientists estimate that the current rate of extinction is 100 to 1,000 times greater than the typical background rate. The extinction rate tends to increase as global temperatures increase. Data suggests that the 6th human-caused mass extinction is likely to occur unless dramatic action is taken. Figure 25.7 shows how the temperature increase favors mass extinctions. One effect from mass extinction is that it can alter the ecological communities and niches available to organisms. When there is a mass extinction, it takes a long time for the diversity to recover, from 5 to 100 million years. The percentage of marine organisms that were predators increased from the Permian to the Cretaceous mass extinction. Mass extinction can pave the way for what we call adaptive radiation. Adaptive radiation is when one species branches out from the ancestor due to the new environment condition and evolves. Adaptive radiation may follow mass extinctions, the evolution of novel characteristics, or the colonization of a new region. After the extinction of terrestrial dinosaurs, mammals underwent adaptive radiation. When the dinosaurs disappeared, it allowed for the expansion of mammals in both diversity and size. Other notable radiation include photosynthetic prokaryotes, large predators in the Cambrian, and the land plants. Figure 25.19 shows the adaptive radiation of mammals. Adaptive radiation can occur when organisms colonize new environments with little competition. For example, the plants on the Hawaiian Islands. The plants on the Hawaiian Islands have been derived from the same common ancestor but just exist on different islands. This is an example of adaptive radiation. Different species branched out from the ancestor due to new environmental conditions and evolved. Let's talk about how major changes in body form can result from changes in the sequence and regulation of developmental genes. By studying the genetic mechanism of change, it can provide an insight into large-scale evolutionary change. So there are genes that program development. They are called developmental genes. They turn on or turn off to trigger when certain things grow and to control the rate, timing, and spatial pattern of changes in an organism's form as it develops into an adult. The rate or timing of developmental events is called heterochrony. It can have a significant impact on the body's shape. For example, the different shapes of human and chimpanzee skulls is a result of different parts of the skull growing at different rates. So you can see that the human and chimpanzee fetus skull look very similar, but have different rates of growth at different times, which forms a protrusion in the chimpanzee adult skull, which is not in the human adult skull. Heterochrony can alter the timing of reproductive development relative to the development of non-reproductive organisms. For example, Pediamorphus is sexually mature species that still have juvenile structures. In Pediamorphus, the rate of reproductive development is accelerated when compared to somatic development, so some of the juvenile structures remain in the adult. Here's an example of Pediamorphosis. The species still has the external gills, although he is an adult. Homeotic genes are one type of gene that is connected to heterochrony. The control of the placement and organization of body parts is controlled by homeotic genes. When certain body parts grow at certain times, it is due to homeotic genes. For example, the basic features determined by homeotic genes are where a pair of wings and a pair of legs will develop on a bird, or how a flower's parts are arranged. The products of one class of homeotic genes is the Hox gene, which are responsible for the position information during development. For example, fins in fish and limbs in tetrapods. So during the Cambrian explosion, there were a tremendous increase in the diversity, and it is still a puzzle why. We know that developmental genes may play an especially important role in the diversity of organisms because changes in the developmental genes are what result in new morphological forms. Changes in genes are what is likely to cause different morphological forms. It is likely that there was some type of gene duplication event that produced new developmental genes, which is how we got new morphological forms. A possible mechanism for the evolution of six-legged insects from a many-legged crustacean ancestor has been demonstrated in laboratory experiments. The UBX gene is a gene present in insects. The UBX gene regulates the decision regarding the number of wings and legs the adult insect will have. Specific changes in the UBX gene has been identified that can turn off leg development. The UBX gene is turned off in the many-legged crustacean, which allowed for the evolution of the six-legged insect. So there are many changes in the developmental genes. So changes in morphology are likely a result from changes in the regulation of developmental genes rather than changes in the sequence of the developmental genes. For example, the three-spined sticklebacks in lakes have fewer spines than their marine relatives. The gene sequence remains the same. but the regulation of the gene expression is different in the two groups of fish. Remember, this was also seen in the insects when the UBX gene regulation has changed, which allowed the many-legged crustacean to evolve into the six-legged insect. Here's an image of the three-spined stickleback found in lakes. They have fewer spines than their marine relatives. And remember that the two species have the same gene sequence, but the regulation of the gene expression is different in the two groups of fish, which causes the loss of spines in the lake sticklefish. Here is the experiment that determined if it was the gene code change that was the cause of the morphological differences, or if it was the regulation of the gene that caused the differences. There was two hypotheses that were tested. Hypothesis A was tested to see if there were differences in the coding sequence of the PITX1 gene. The results showed that the 283 amino acids of the PTIX1 protein in both fish are identical. Then hypothesis B was used to see if there was differences in the regulation of the expression of the PITX1. The results showed that the PTIX1 is expressed in the ventral spines and the mouth regions of developing marine sticklebacks, but only in the mouth region of the developing lake sticklebacks, causing a lower number in the spines of the lake stickleback fish. Evolution is a process where new forms come from changing the existing forms. So most of the complex biological structures we know of, we believe, have come from other previously existing structures. So complexity is built off of things that were previously less complex. Natural selection improves on a structure in a context of its current utility. So structures evolve in one context as it is chosen for a different function. For example, complex eyes have been evolved from simple photosynthesis cells independently many times as they were chosen for different functions. So if we look at the eye structure of different organisms, we can see how the eye starts off less complex in an aquatic snail and then becomes more complex in each species as it is chosen for a different function. And it continues to evolve into the complex camera type eye evolves, which is in squids and is very common to the vertebrate eye, which is like ours. So the last thing, let's talk about evolutionary trends. Fossil records can be misleading when trying to extract a single evolutionary progression. Apparent trends should be examined in a broader context to fully understand what is happening. Now, the species selection models suggest that differential speciation success may determine evolutionary trends. It is important to know that evolutionary trends do not imply an instinctual drive towards a particular phenotype.